Hey, hey, hey! This lecture is designed to help you with the 202 exam, the first exam. So let's go ahead and jump in. Hopefully you've already watched the endocrine video and you're starting in on blood. Okay, well, first, what is blood? Blood is, you want to highlight a connective tissue. And you probably remember that, from 201. But what it does is it transports oxygen around. It transports from the last lecture. It transports the hormones around. It also maintains the stability in the interstitial fluid that is, swelling in the fingers and joints and all that. Or it maintains that fluid. And it moves the heat around. So that's what it does. How much do you have? And that's just, average. Obviously, the bigger you are, the more blood you have. But blood is about 8% of your body weight. And then you want to know that last part, the 3 types of blood cells. There's red blood cells. We'll call those, from this point forward, you need to know that a red blood cell is an erythrocyte. Okay? A white blood cell is a leukocyte. The prefix ERYTHR means red. So an erythrocyte is red. A leuko, L-E-U-K, means white. So like leukemia, leukocyte, that's white. And then platelets, those will be called thrombocytes. And so I think you know this, but make sure, okay? Red blood cells, they're for carrying oxygen. That's the purpose. They carry oxygen. White blood cells, they are immunity. They help you fight off diseases. Platelets, what they do is they stop up clot. They make clots. They help you scab, or they plug up when you're bleeding. And so those are the 3 types of cells. And there's different white blood cells that we'll talk about in this chapter. And so that just kind of shows you about how much of everything. Like how much plasma, how much platelets. And plasma is just the liquid. And then how much red blood cells. Okay, this is just interesting that the book shared. These are precautions that you would use no matter what in the hospital. And so you would always take these universal precautions in preparation in case you don't even know the patient. Or maybe, you have no idea if they have a blood disease. But you would always wear gloves, use the appropriate place to throw away needles, and then wash your hands. So that's just universal precautions for everyone. Okay, so red blood cell, we already said, it's called an erythrocyte. They are, you want to know all of this. Oh, man. Okay. They have biconcave disc shape. They're 1/3 hemoglobin. Hemoglobin is a protein that allows them to carry oxygen. So hemoglobin is the protein that allows them to carry oxygen. Deoxyhemoglobin would be without, oxyhemoglobin with. They don't have a nucleus. So they do have a nucleus when they're really young, when they're first become cells, but then after that, the nucleus is gone. They don't do mitosis. They don't divide. They, you'll remember from some previous science class, maybe mitosis is the division of cells. Well, red blood cells are created in the red bone marrow, and then they go to the spleen to die. So it's not like they divide and divide like skin cells or muscle cells or bone cells. They are born and then they die in the spleen. And then they can produce ATP through glycolysis, but they don't do all of cell respiration. So like they don't have to have food particles. They have enough energy because they don't live for very long. And then this is really just kind of average, kind of interesting. The number of red blood cells do you have? And so that's the numbers. Okay, highlight this. Erythropoiesis is where the red blood cells are made. So erythropoiesis, red blood cells are made where? in the red bone marrow. So make sure you've got all of that. This is a negative feedback mechanism. What that means is when you're low on blood, you make more. When you've got enough blood, you don't make as much. So you got to keep that normal amount. Homeostasis, negative feedback. Remember, those two words are hand in hand. We regulate homeostasis with negative feedback. And so erythropoiesis, you want to know that for sure, is red blood cell formation. Okay? Red blood cell formation. Where? In the red bone marrow. Erythropoiesis. Okay. Okay, so anemia is next. I knew there was something funny. Okay, back a slide, sorry. The dietary factors that would affect your blood. Well, we all know about iron, which is anemia, right? But some other things, vitamin B12, and you get that from eating red meats, like meat, steak, hamburger, such like that. Iron, and then the folic acid. And so all three of those, B12, usually, if you have a deficiency, usually result of damage in the stomach lining. Iron, nutritional requirements. Women need more iron than men, and that's why more women are anemic. Vitamin C increases the absorption rates, and then folic acid you get from eating grains and cereals, DNA synthesis. And so the folic acid, I know that's a big deal. When you're pregnant, you take the vitamins with folic acid in them to help with, it says, development of the fetus during brain development. So if you don't take the folic acid right, the brain won't develop appropriately. Okay, types of anemia. Read over all these. You want to probably make a card for each one. But two you want to pay really close attention to. The first one is hemolytic anemia. Toxic chemicals. Red blood cells are destroyed. And then the second one is sickle cell anemia. And that's where the red blood cells they're shaped funny. They're shaped like sickles instead, like a moon. I think maybe I have a picture later, but they're shaped like a moon. And because of that, they don't have as much hemoglobin. So they don't carry as much oxygen. And so people with sickle cell, it's a recessive disorder, so you can be a carrier. People with sickle cell, they may have immune system problems, and they get sick more often, and they're sleepy, but it's because of the oxygen in the shape of their red blood cells. Okay, besides anemia, here's some more red blood cell disorders. You got the first one, micro means small, microcytosis, micro small. You have an abnormal number of small red blood cells. Hypochromia, hypo means below, so HYPO means reduced chromia. Anisocytosis, excessive inequality of size, so some big and some small. Poikicytosis, abnormal number of large. And then make sure you highlight this last one. Cyanosis is a blue or purple coloration of skin due to low oxygen. So, I'm sure you've seen babies when they start to cry and they're holding their breath, their face will turn blue. So that would be cyanosis. So, blue or purple coloration. Okay, life cycle of a red blood cell. So we already know they're made in the red bone marrow, right? This just kind of goes through the steps. So the first one, you got to have nutrients. The blood transports the absorbed nutrients. We've already highlighted, but you could do it again if you wanted. They're produced in the red bone marrow. Red blood cells circulate in the bloodstream for about 120 days. So that's about how long they last. Macrophages, those are white blood cells. They eat them. They phagocytize them. And then they start to break down. The iron is recycled, and that's interesting. And then the bile is secreted into the small intestines. So that just kind of goes through the steps of a red blood cell. And that's it for red blood cells. White blood cells, however, there's 5 different ones that we're going to talk about. Okay, so what does a white blood cell do? That is your immunity. So it protects against disease. Highlight that. If you get the flu, or if you have a cut, an infection enters the body, the white blood cells are the cells inside your body that fight, right? So think about them like little people of an army inside you. And you have an amazing immune system. And we're going to talk more about the immune system later in the course. But for now, we're just going to talk about the white blood cells. But God, immune system is one of my very favorites. Okay, they are also produced in red bone marrow. So not yellow bone marrow, but red bone marrow. And they are under the control of hormones. Imagine hormones controlling something after that last one, right? Okay, there's 5 different kinds. And they divide them up into 2 different ways. There's the cells that look grainy, or in my opinion, I would have said sandy. Granulocytes, so just grainy or sandy. So they have a granular cytoplasm, and that's going to be the neutrophils, eosinophils, and basophils. Okay, agranulocytes, the prefix A means not. So under a microscope, these don't look grainy or they don't look sandy. They look smooth. Lymphocytes and monocytes. So you want to know the difference. I might ask you a question something like, which of the following is not a granulocyte? Neutrophils, eosinophils, basophils, or lymphocytes? And you're going to choose lymphocytes. Okay, this picture, I just found it on Google and thought it was nice. So it shows you, it's in the bone marrow. Whoops. There we go. Okay, in the bone marrow. And then you have the erythrocytes. That's the red blood cells. We already know about those. And then there's the 5 different white blood cells. And see how these are like sandy looking, like grainy looking? Okay, that's a basophil. Then the neutrophil is the biggest one. Eosinophils, that one. A monocyte is that one. And then a lymphocyte is small, however, very important. And so, it shows you right here, basophil helps with inflammation, so swelling and sinuses and all that. Neutrophil, eosinophil, and monocytes, they phagocytize, and that means to eat. Like I think about like Pac-Man, they go around eating the germs. And then these are all leukocytes. They're all white blood cells, right? All those are white blood cells. And so I just thought that picture was neat, kind of showing you. Okay, first one is neutrophils, and there's a reason it's first, because it is the first to arrive. Whenever you get an infection, let's say you get cut on your finger, the neutrophils show up first, ready to fight. And so they are elevated. Think about them like the first line of defense. They're elevated during bacterial infections. So if there's normally 54 to 62, and you got to know the percentages. So you take a blood sample. Somebody's neutrophils is up to 75%. Boom. It's a bacterial infection. So these percentages are important. So of the blood, 50 of the white blood cells, 54 to 62% is normal range. And if it's above that, then that would indicate a bacterial infection. Okay, looking at it, it says it has a lobed nucleus. That dark in the middle is the nucleus, is lobed. They are phagocytes, which means they eat. They walk around like Pac-Man, chomping at the bad stuff. And then it tells a description of them. But they're the first ones to show up. You also want to know, not only first, but you want to write in, they are the greatest. So that's the highest percentage. 54 to 62 is the highest percentage. The rest of them, what's over half? Obviously, it's the highest, right? So the rest of them are not that high. Okay, the next one we're going to look at is eosinophils. They have the granules. They look sandy, right? They have bilobed. They do moderate the allergic reactions. Whoops. So let's highlight allergic reactions. This is what you want to highlight for sure. They defend against parasitic worms. So if you take a blood sample and it's higher than 1 to 3, so let's say it's 5%, that would give you an idea that it's either an allergic reaction or a parasitic worm. And if I was a doctor, I'd probably assume allergic reaction first, right? But allergic reaction and allergies. Allergies and parasitic worms. Okay, so 1 to 3%, parasitic worms, and allergies. The next one is basophils. Large, sandy in appearance granules. They release histamine. Okay, and what that does is it stimulates inflammation. So we've probably all taken an antihistamine, like an anti-allergy pill. Okay, so it releases histamine to stimulate swelling. So that would stop the spread. That's the purpose of the swelling. It also releases heparin to stop the blood from clotting. So you want to know that histamine is inflammation and heparin is what prevents blood from clotting. It's very small percentage, less than 1%. So not very high percentage at all. So, but you want to know histamine and heparin. Basophil, histamine, heparin, 1%. Okay, monocytes, they're the biggest ones. They are in a percentage of 3 to 9. They phagocytize bacteria, dead cells, and then just dirt, grimy stuff, that gets in like in a splinter or whatever. So those are the monocytes. And monocytes become, there it is, they become macrophages. And macrophages are big, P-H-A-G-E, big eaters. They go around like Pac-Man eating all that's bad. So monocytes become macrophages, 3 to 9%. They phagocytize bacteria and the dead debris cells. Okay, lymphocytes. These are my favorites. 2 types of lymphocytes. These are not grainy, right? You can see that in the picture. They are A granulocytes, but there's 2 types. There's T cells and there's B cells. Okay, T cells, what they do is they release a toxin that destroys the pathogen, right? So they release, it's like almost like they drill holes, like they have guns and they shoot it with a little toxin. So T cells directly attack the pathogen. B cells produce antibodies, and the antibodies, antibodies are proteins, they're shaped kind of like that, and they attach to the pathogen, directly to the pathogen, and they inactivate it so that it can be found in the spleen. And so lymphocytes, there's T cells and B cells, and there's directly attack and produce antibodies. And we said these are agranulocytes. They don't appear as sandy under a microscope. They aren't grainy. Okay, so what I would do now if I were you is I'd take a little break and think about draw a table, something like this, the name of the white blood cell, the percentage, and what special features like parasitic worms or whatever else I told you to highlight. So there's 5 of them. And I'm sure your drawing will be better than mine. But the last one we covered was a lymphocyte. Monocyte. And I would fill this out. So neutrophils, that's the greatest, remember. You got 54 to 62. Eosinophils, that was 1 to 3%. Basophils, they're less than 1. Monocytes, I believe, is 3 to 9. And lymphocytes is 25 to 33. Okay, so in those percentages, I know they're numbers and you don't want to memorize them, but they're really important because of what they mean. And so if you took a sample of white blood cells, they actually count them. They count the number, and now there's a machine that does it, but it used to be a person back in the day. So there's a machine that counts and says, of the white blood cells, there's 54% that are neutrophils. That's good, right? And this is elevated in bacterial infections. Eosinophils, we said worm and allergies. So you want to make sure that you know this. There's a couple of questions on the test about these white blood cells. So make sure that you go over this and fill it out. This one has the heparin and histamine. Bacteria. Dead cells. Monocytes, you also want to know they become macrophages, right? And then lymphocytes, that's T cells and B cells. And B cells make antibodies. Okay, so take a second and see if you can fill that out without me. Maybe try it again on another sheet of paper. Memorize that real good. And then move on. Okay. Oops. What do the white blood cells do? We already talked about phagocytosis, and that's a really, really important one. First, we'll talk about diapetesis. That's where the white blood cells can squeeze through. They can squish and squeeze through capillary walls. They can leave the red blood cells and go towards the infection site. So it always reminds me of like how a rat would get into a garage or something. It shows the picture down here is what that is. It shows the white blood cell squeezing through that little crack. You see it squishing through? It can leave the capillary, the artery or the vein. It can leave the vessel and actually go to the infected site. So like if you got cut on your finger, it could leave and travel towards the skin. Phagocytosis is the engulfing. Think about it like Pac-Man. Engulfing and digestion of pathogens. Neutrophils and monocytes are the most important ones. Neutrophils, that's the first ones. Remember, monocytes become macrophages, and they're really important. Okay, inflammation, that would be where the reaction, it restricts the spread. So like the capillaries, the arteries and veins, they would close up so that the infection would stay in that one spot. So swelling, so inflammation or swelling. And then positive chemotaxis, really interesting. You want to know this whole slide. I would have a different index card for every one of these if I were you. So 4 different index cards there. Write positive chemotaxis on the front. On the back, write attraction of white blood cells to an infection site by chemicals released by damaged cells. So, okay, you cut your finger. The skin cells at your finger release a chemical that says we're damaged, we're damaged, we're damaged. The white blood cells can travel towards the damaged cells, which is so interesting that they go that way. They go towards it. Okay, so this is just showing you the pictures. Neutrophils do respond to the bacteria. They're the first ones to get there, right? They run to the infection site. So looking at this picture, you get a splinter. Okay, the splinter is covered with bacteria. The bacteria is green, so the bacteria are there. Now start on 4. The injured skin cells, it says, injured cells release histamine, causing the red blood cells to dilate and become leaky. So the blood vessels will open up and allow the white blood cells to go there. The neutrophils get there first. And then the neutrophils destroy it by phagocytosis. And do you remember what phagocytosis is? That's the big eaters, Pac-Man. Okay, here's just a count here. White blood cells should be between 3,500 and 10,500, depending on how big you are, right? So a procedure we can do that counts the white blood cells if you have less or more. Okay, so leukocytosis is first, and that's going to be higher than 10,000. Leukopenia is going to be less than 3,000. So white blood count is high or white blood count is low. If the white blood count is high, it could mean infection or vigorous exercise or lots of fluid lost. You want to know those numbers and everything. Leukopenia, low, P-E-N-I-A means small. Low, it could mean typhoid fever, flu, measles, mumps, chickenpox, AIDS, polio, or anemia. So that's high and low. And then when you have a differential white blood cell count, that's where we were talking earlier. You know, if you have 50% neutrophils, or, you know, then you have a small number. Or if you have 70%, it's a high number. So that's a differential white blood count. And they have machines to do it now. Neutrophils generally indicate bacteria. I do want to highlight this. We haven't talked about it yet, but, okay, of those lymphocytes we talked about, there's one called T cells. There's a specific type of T cell that we'll talk more about when we get to immunity called a helper T cell. Helper T cell, that's the type of cell that's infected during an HIV contraction. So if you've got HIV, it's a virus that infects the helper T cells. So just be able to associate HIV and helper T cells for now. Okay, so that shows you exactly what was in that table that we just drew out and kind of went over. Leukemia, we've heard of leukemia. Probably all know someone who's suffered from that. Leukemia is cancer of the L-E-U-K, means white. So leukemia is cancer of the white blood cells. And there's two different kinds. If you have acute, it shows up fast, lots of high pain. If it's chronic, it's slow and maybe undetected for even years. There's lymphoid leukemia, and that's cancer in the lymphocytes, right? The lymph nodes. There's myeloid leukemia, and that's cancer of the granules, the sandy looking ones, right? It's produced in the bone marrow. So that just kind of gives you some information about leukemia that was in the book. Okay, platelets, those are your clotters. Platelets, also called thrombocytes. That's what makes it clot, right? Stops the bleeding. So highlight that word. Help in hemostasis, stop bleeding. So know that hemostasis is stop bleeding by stopping up the damaged blood vessels. Okay, and this table is really a nice summary of everything we've gone over. So it talks about red blood cells at the top and, you know, their description and numbers. So this is just a summary. The 5 types of white blood cells, the neutrophils, eosinophils, basophils, monocytes, and lymphocytes, and then the platelets, or thrombocytes. And what they do, they fragment, they control the blood from bleeding, right? And so this is just, look over this real good. You probably already got it on your note cards at this point. So you don't have to rewrite this, but just look at it. A nice summary there. Okay, what makes the blood liquid? So far we've only talked about cells, but the liquid part is the plasma. It's clear or kind of a yellow. Well, I guess they call it a straw color in the book, but it's kind of a, you know what the color of blood fluid is. You've seen it. And so it's a clearish color. It is the liquid portion. It's actually over half of what we call blood is plasma, and it is 92% water. So it's mainly water, but it helps regulate the fluid and the electrolyte balance, and so it moves the fluids around. The blood's got to be liquid, right? Okay, there's a picture there. You can see the different, in the plasma, there's proteins. Now, I want to point out some that I want you to focus on. Albumin is one. It's kind of like egg white. It's pure protein. 60% of the blood plasma proteins are albumins, and they help maintain the colloid osmotic pressure. So that's a pretty important one. It's a high percentage. And the other one is fibrinogen, and it helps to clot. Sometimes you want to clot when you want to plug up something that's bleeding. So fibrinogen, we'll talk about in a second, helps with blood coagulation, or coagulation means clotting. Okay, so what does the blood move around? It moves around the oxygen and the CO2, the carbon dioxide, and you breathe in. Oxygen goes into your lungs, and then you got to get oxygen all the way out to every single muscle, even the ones in your big toes, right, and little toes. Okay, and then there's nutrients. Okay, amino acid, just really for future reference, or for just so you will know it. Amino acid is a broken down protein. Sugars, like glucose, nucleotides, that's DNA. So DNA is made up of nucleotides. Proteins are made up of amino acids. And then the lipids, those are all going to be fats. Triglycerides, that's the oily fats. Phospholipids, that's, what's in cell membranes. And then cholesterol would be more like a solid fat, like bacon grease. So all of the fats, this is what the blood moves around. So it moves around nutrients and it moves around gases. Okay, on this next slide, non-protein but still got nitrogen in them. So molecules that have nitrogen but they're not proteins. So we talked about like the albumin and the fibrinogen. Those are proteins. But urea, that's going to be when you break down the protein. The word catabolism means breakdown. So product of protein breakdown, or catabolism, is most of it. And that's when I think about urine, right? Urea. And then uric acid, product of DNA catabolism, so DNA breakdown. So urea is protein breakdown. Uric acid is DNA breakdown, right? Nucleic acid. And then the amino acids is also protein. Okay, creatine is what stores energy, and some people will take creatine supplements. And creatinine is a product. You get creatinine when you break down creatine. But the amount of nitrogen in the blood, so the blood urea nitrogen, could indicate whether the kidneys are working. If it's too high, then the kidneys may not be excreting normal amounts of urine. Too low could be renal function. So that's just kind of telling you nitrogen, other than protein, nitrogen in the blood. Okay, if you ever drank smart water is supposed to be water with electrolytes. And we pay extra for it, but we probably all have enough electrolytes unless we, are sweating profusely. But electrolytes are salts mainly, but they're pieces in the blood. What's the word I'm looking for? Elements. Elements in the blood, like sodium, potassium, and you can see them listed there. Sodium and potassium are the most important ones. So they're in the water. So they're absorbed from the intestines or released as a byproduct of metabolism. Now here's the ones found in the blood. Sodium, think about sodium being... So think about sodium being the most important electrolyte. So you've got the, Na+, you remember that from some class before, I think. Right, that's the salt levels. And then potassium, calcium, magnesium, chloride, bicarbonate, phosphate, and sulfate. So take a second and maybe try to list those, practice with them. Of course, sodium, potassium's important, and then chloride is really important. So those are the three main ones that you want to maybe highlight and take note of. But we all need electrolytes, and I always think, I like smart water. I think it tastes a little bit better. But You know, as long as we're eating a well-rounded diet, we probably don't need smart water. But it tastes good, so I buy it. Anyway, okay, the next slide talks about hemostasis, refers to clotting, right? When you have a cut and it's bleeding, how do we, hemo, H-E-M-O, means blood, stasis means stability. So how do we get the blood to stop bleeding so that we don't lose too much? And we know we got the thrombocytes, right? The platelets. So hemostasis, highlight that, refers to stoppage of bleeding. Actions that limit or prevent blood loss include, so the blood vessels will start to flex, spasm, flex, relax, Okay, platelet, you got to have, the platelets are going to plug up the bleeding area, so that's kind of like a cork in a wine bottle. They're going to plug it up. And then the blood is actually going to coagulate, and that word means to stick together. So these mechanisms are most effective in small, like if you have a little small nick in a blood vessel. I mean, not so much if you have like a 3-inch cut on your arm, but a little small nick in the blood vessel. And so those are your 3 ways for hemostasis. And remember, hemostasis is the stability of blood, right? To get the blood to stop bleeding. Okay, so now we have a slide on each of them. What is a vascular spasm? So that's going to be where it flexes. So smooth muscle in blood vessel contracts rapidly, like, you know what spasms are. Slow blood loss very quickly, and ends of vessels may close completely. So they flex, relax. And it's going to be stimulated. It says triggered by stimulation of blood vessel wall pain receptors. So the pain receptors stimulate it in the blood vessel. Okay, the next one is platelet plug. Most interesting thing ever. And that's how it's kind of like a cork. You can see the thrombocytes, how they plug in. So platelet plug, triggered by exposure of platelets to collagen, and that's in your skin. But collagen is a protein that helps skin be firm. Platelets adhere to rough surfaces and form a platelet plug. Okay, highlight this. It's going to be when there's a break in the vessel. So maybe write it in, break in vessel. It formed in response to blood being exposed to collagen. So highlight that word too. Formed in response to blood being exposed to collagen. And then blood coagulation. That's where the blood, it clots together. Y'all look at that, how it's like a spider web on top of it. It's a protein that makes it all spiderwebby and makes the blood stick together. So blood coagulation is the clumping of the blood. Most effective mechanism of stopping the bleeding, hemostasis, initiated by 2 different methods. It's either triggered by the outside the blood or inside the blood, extrinsic or intrinsic. Vitamin K is necessary. Major events in conversion of soluble fibrinogen to insoluble threads of fibrin. So highlight that. You're going to, or maybe just write it in and make it a little bit more easy to read. That's a protein. Fibrinogen to fibrin. So that's what this is. The yellow spiderweb stuff that you see. You got to make that protein fibrinogen that's produced immature form become the active form. And with blood coagulation, we're going to look at, there's the 3 in a nice table for you, organized. Okay, there's extrinsic and intrinsic. Now, extrinsic is when it's triggered outside the body. Come on, EX, you know that means extrinsic. Okay, you want to highlight the word thromboblastin. So damaged tissues release outside the blood, release a hormone or a chemical molecule called thromboblastin. Right, so it's not in the blood. This will be outside the blood when there's a cut that's not in the blood vessel, not a small cut in the blood vessel, but somewhere else in the skin, maybe, and they release thromboblastin. Thrombin is going to be, we already highlighted this, fibrinogen to fibrin. Okay, and that's what this mass is called a blood clot. So that fibrin, that on that last slide, what you saw, that yellow, that's a blood clot. Okay, and then extrinsic and intrinsic, they're both positive feedback. So extrinsic and intrinsic are both positive feedback. So just make sure you got a couple things on this slide. You've got that extrinsic is outside the blood. Okay, you've got the tissue thromboblastin, and really what it is the thromboblastin is released when it comes in contact with something other than what's in the blood. So when it comes in contact with, maybe write this down, something like calcium. Thromboblastin is what converts fibrinogen to fibrin. The fibrin, that's the spiderweb looking stuff, the yellow stuff from the last picture, sticks to the damaged blood and makes it like a clot. So it makes it coagulate. And we already said they're both extrinsic and intrinsic are both positive feedback. Now, positive feedback. Intrinsic. And so intrinsic we would know would be inside the blood, right? So can start without tissue damage, so it's inside, intrinsic. Activated when blood comes in contact with a foreign surface, such as, it says collagen there, but we're going to say, such as that calcium, right? But collagen too. Triggered by, extrinsic was thromboblastin. Intrinsic was the Hagman factor. That's just the chemical messenger that starts the process. So, inside the blood. And so make sure you got kind of a summary here. Extrinsic was thromboblastin. While intrinsic is Hagman factor. So chemicals inside the blood triggers coagulation. So inside. Both of which are going to be positive feedback. So just a table with a summary. We know both of these things. Oops. And then we know thromboblastin and Hagman factor. So kind of focus in on those. Okay, what happens to a blood clot? Well, we know blood clots are good when they stop the bleeding of vessels, but they're bad when they stop the flow of blood, to get to the muscles and all that, or to the brain or heart. And so small clots just disappear. Okay, there's an enzyme called plasmin that actually digests that spiderweb fibrin. And dissolves the blood clot. So plasmin is the enzyme. Now there's two types of blood clots that we'll focus on. A thrombus is an abnormal blood clot that forms in blood vessels. So you want to know that word. Highlight that. And I do that all the time. An embolus is a blood clot that's moving. So embolus, moving, is a blood clot moving through the blood vessels. Thrombus is an abnormal blood clot that forms in the blood vessels. So highlight both of those things. Okay, just some vocabulary. Thrombosis is a blood clot in a vessel supplying a vital organ. So that's bad, right? When you have a blood clot in a vessel that's supplying blood to like the brain or the heart. Okay, infarction, you want to know that word. That's when the tissue actually dies. So death of tissue, because it's not getting blood, it's not getting oxygen. So death of tissue, which has blocked the blood vessel due to the blood clot. So the blood clot is preventing oxygen from getting there. Embolism, blood clot that travels and then blocks a blood vessel. such as a pulmonary embolism. So it was traveling and then it got wedged and stopped in the lungs. Atherosclerosis is the accumulation of fats. That's going to be high cholesterol. And we'll talk about cholesterol in another chapter. But accumulation of fat in the lining and sometimes can cause a blood clot. So in the arteries, in the arterial lining, common form of thrombosis. So kind of know all those are kind of like vocabulary words. These are all clot busting drugs, or drugs that are taken to get rid of blood clots. The one I really want you to focus on is this TPA, tissue plasminogen activator. It's got to be given within 4 hours of the heart attack. So it's given quickly and helps restore the circulation, so it gets things moving again. And the rest of these are just other things to get blood clots. One thing I think is interesting is the medicinal leeches. I know we've probably seen those on movies, how they use the leeches to suck the blood. Isn't that just interesting? But it can be used to prevent clotting. Okay, next slide. Talking about blood types. So here we are. This part's really interesting and very applicable. But all red blood cells, well, not all of them, but A and B have antigens on them. So an antigen is any molecule that evokes an immune response. An antigen is basically just a surface protein. It's not bad. It's good. So it's on the surface of the red blood cells. So think about, here's your red blood cell. And you have surface proteins called antigens on them. And they may start an immune response, but they're normal to you, right? They may start an immune response if someone else had those, but those are yours. So if the immune system finds a foreign antigen in the body, it produces antibodies against the antigen. So your red blood cells have your antigens, and that's good. That's normal. Your immune system is that's normal. Okay, but if it finds a foreign one, like let's say it finds an antigen that's not shaped like a circle, then that would start an immune response. Okay, antibodies are proteins that react against a specific antigen. And so antibodies are proteins. They're shaped like that, but they're way smaller than red blood cells, so that's not a very good diagram. There we go. They're kind of shaped like tongs that you would use on a grill or something. But they're proteins. They're made by those B cells, the B leukocytes. And what they actually do is so now the antibody will be green. They connect to the foreign antigen. So antibodies react against the antigen. They connect to it. So it says, in a compatible blood transfusion, in an incompatible. So you have A blood and you got B blood transfusion. The donor red blood cells start an immune response in the recipient. So I have A blood, but I got B blood transfusion. And then the antibodies are going to all attack that foreign blood. And that would be bad. The antibodies, what they do, they attack it like this one shown here. They attack it, and then it starts to all clump together. So it makes a big clump of the wrong red blood cells and antibodies. So it says, evoke an immune response in the recipient and antibodies in the recipient's plasma. Coagulation, that's clumping the donor's red blood cells. Okay. Agglutination, that's going to be clumping, which occurs when the antibodies encounter specific antigens. There are 33 antigens on red blood cells, but we really only care about A and B. Only the antigens of the ABO and RH, we care about that one too, group evoke serious transfusion reactions. So there's 33, but we're really focused on 3, A, B, and RH. O is not really one. Okay, so ABO blood group. You want to draw this table out, pay close attention to it. It's based on the presence or the absence of 2 major antigens. There's antigen A and antigen B. Okay, if you have A blood, then you have A antigen. So here's your A blood and you have A surface proteins. right? That's good. That's normal. It's fine. Now, if you were, that means that your body naturally makes an antibody against the B's, which is the other option. If you have B blood, then you have B antigen. Which would mean if an A were to get into your blood, it would start an immune response. Okay, if you have AB, you have both. So you have A's and B's. If you have O, that means you have a little naked red blood cell. No surface proteins. Both anti-A and anti-B. So let's just look with me here. If you have A blood, so just look at that one. And a B antigen comes in your body. Then what's going to happen is you're going to have an immune response, and antibodies are going to attack it and cause it, those are antibodies, the red is antibodies, and cause it to all clump together. And so that would be an immune response. Okay, I know my picture was better, but the book provides a picture too. So you can see here, this first box right here, that's A blood, and look like it has little bitty orange antigens on it. A antigens, which are surface proteins, and then you can see the antibodies are all clumped together. I was just drawing one at a time. The B, see how the antigen is shaped differently? Look at comparison here. The B antibody, the B antigen, and the A antigen, they're shaped differently. And to your immune system, they feel. Immune system cells feel around, and when they feel something abnormal, they attack it. If you have AB, you have both. And then if you have O, you have naked red blood cells. When red blood cells come in contact with antibodies against them, they will clump. Okay, O, because it's naked, it wouldn't start an immune response. So if you had let's say you had AB blood or A blood or whatever, you got O red blood cells, well, there's no surface protein, there's no A or B to start an immune response. So for that reason, we call O the universal donor. AB, we say, okay, if you have AB blood, look at this. Then, if you got A blood, it'd be fine. You're used to A's. A's are normal. If you got B blood, that's fine. B's are normal. You've got A's and B's. So we call that the universal recipient. So O is really generous. AB is really selfish. And so this table shows you transfusions. So take a second and look at that and make sure you understand it. But you want to know that O is the universal donor and AB is the universal recipient. Okay, what about positive and negative? We got A, B, and O. Hopefully we understand that. What about the positive and the negative? Now, it actually is antigen D. Whoops. There we go. RH positive would be antigen. That means you have A, like let's say you're A positive. That would mean that you have A antigens, but you also have another antigen called antigen D. You have A negative doesn't have that antigen D, A positive does. But we call it the RH positive or RH negative. And we call it that because it was discovered in the rhesus monkey. Let's see if that says, does it say that anywhere on the slide? I don't guess so, but it's because it's named after a monkey, the rhesus monkey. So if you got it, you're positive. If you don't, you're negative. So they don't really call it antigen D. They call it RH positive and negative. Anti-RH antibodies form only if, so you only have the antibodies against the RH in response if you've gotten a positive. So if you have positive blood, then you can receive positive or negative blood, right? Because the negative the absence of antigen D isn't going to start an immune response. It's foreign molecules that start immune responses. And so if you're negative and you got a positive, then that would cause a response. So 85% of the population are plus, so most of us are positive. The seriousness of the Rh is important when we're talking about pregnancy. So erythroblastin, or I learned it as hemolytic disease. So that's what's on the lab activity. Erythroblastosis or hemolytic disease. Okay, so let me tell you the story. You get RhoGAM is the drug. You want to highlight that. You want to know the drug. And you want to know hemolytic disease in newborns. But here's what happens. If she's pregnant and she is Rh negative, so I'm going to put a negative sign in purple on her face because you want to remember the story. If mom is negative and baby is positive, right, the baby's blood and the mama's blood doesn't mix. So if mom is negative and the baby is positive, that's fine. It got the positive from the daddy, the sperm donor. What happens when the baby is born, see, the baby's got the antigen D. What happens during childbirth, cells from the Rh positive fetus may enter the mama's bloodstream. So when it does, when the mom gets some positive, she's negative, remember, but when some positive enters, it forms these antibodies. No big deal. Mom's exhausted. Who cares? She's really tired anyway. Okay, when she has a second baby, if the second baby is positive, remember, mama's negative and she got antibodies. I don't want you to be confused by that little line. Okay, so mama's negative, she's got antibodies. When the second baby is born, so second child, the antibodies will attack the baby because it's a foreign. It's like, you have antibodies against certain diseases, and when that disease comes in, your immune system attacks it. So in the next Rh positive pregnancy, so the next one, the second pregnancy is when it's a problem, maternal antibodies attack the fetus. And so what they do is, after you have the baby, first baby, if you're negative, mama's negative, baby's positive, they give you what's on that last slide, the RhoGAM drug. RhoGAM drug. So we highlighted this. Oh, we went back too far. The RhoGAM drug given to mom, last bullet. So her immune system is not activated. And I think, y'all Google it and make sure I'm right, but I think they just give it to the mom if she's negative. Alright, that's it. I hope that was helpful. Make sure that you've watched both videos to study for exam 1.